we're trying to build a platform that enables end-to-end workflow. So from upstream cell preparation to our core technology, which is our single-cell isolation and molecular barcoding technology, all the way through providing different options for library preparation and looking at different analytes and through data analysis. So most of you are familiar with BD Rhapsody. This is our microwell array-based single-cell capture platform. So it uses these microwells to physically partition single cells, and the cells are lysed in the wells. And we have, as Simone mentioned, we have the instrument here for you to take a look at later. But what's really cool about this technology is that because it's done in a clear flow cell, so you can really visualize the single cell capture, and that gives you the ability to QC your single cell capture before going into library prep and sequencing. And as you all know, a lot of the cost in single cell experiments is the reagent for library prep, but also for sequencing. So knowing whether you captured 3,000 cells versus 10,000 cells is really helpful to know how you want to allocate your sequencing reads. And then we use solid magnetic barcoded beads, which is slightly different from some of the droplet-based methods. So these beads can be retained for later experimentation. They're also quite sturdy, so you can do a lot of manipulation on these beads, which really expands the flexibility of the assay. And then this entire workflow is done on just a benchtop equipment. So there's not a lot of worry about clogs and things breaking down. If the power goes out, you can still do your single cell capture. We've also developed, spent a lot of time looking at different ways to look at RNA signal, and I'll talk a little bit more about that later. But another exciting area that we're spending a lot of time in is data analysis. So some of the earlier challenges of the single cell technology adoption is data analysis. So you're generating so much data, especially with multiomics, there are different ways to look at it. So the real challenge is to be able to allow the biologists and immunologists who are doing and designing experiments to also analyze their data. And so with the acquisition of SeqGeek, we've been working with the team there to really expand the capability of SeqGeek, which is their single cell visualization tool. And then we've also spent a lot of time looking at other like multiomics techniques that allow us to also probe protein expression. So that's basically our oligo-conjugated antibody technology, where we tag antibodies with a oligo to allow these oligos to be captured alongside mRNA during the lysis step. And we've applied this technology in two different assays. The first is the single-cell multiplexing kit. So this is using one universal antibody that's labeled with one of 12 sample-specific barcodes, and allows you to pull multiple samples into a single run. And it's nice because it reduces the technical work, but also the technical errors that you might see from run to run. And the other application is BD AppSeq, which we launched this past summer. And this basically is a way to allow protein profiling along mRNA expression profiling. And because DNA barcodes are not limited by the spectrum, so you can easily design tens of thousands of unique barcodes. So this allows us to do very high parameter analysis. And I wanted to show you just a couple of data that we generate internally, trying to look at how we can apply this technology. So this is, we obviously spent a lot of time comparing AppSeq performance to flow cytometry, which is the golden standard. And so in this experiment, we basically took CD8 T cells. We in vitro cultured them across over 2 weeks, and then took samples from different time points. And so here, this is comparing flow cytometry data to BD AppSeq data and looking at fold change relative to the starting point, which is fresh, naive T cells. So you can see that for the markers that I'm showing you, the fold change and kind of the dynamic range that you can see with BD AppSeq versus flow cytometry is really similar. And another benefit of including protein expression data in your single-cell RNA-seq data is the ability to have better defined clusters and populations. So you might know that. So I guess this is showing the same data set that I just talked to you about in the previous slide. and circled in red is the fresh cell. So these are mostly naive T cells. And you can see that with only mRNA alone, you don't get a lot of definition of the subpopulations. And this is mostly because naive T cells they're quite small. They have pretty low mRNA abundance, and they tend to be transcriptionally similar across the different subpopulations. But when you add in the protein piece, so these are markers that are more highly expressed, and a lot of them, especially the primary markers, tend to have binary expression, which really help define the different populations. So you get much better definition and resolution of your populations, which allow you to better integrate the data set. Another really cool application is using it to look at mRNA versus protein kinetics. So there's been a lot of past studies and recent studies that show that protein and mRNA expression don't always correlate. So it's highly dependent on the genes you're interested in, the cell state, the cell type. So this is an example of the same experiment where we looked at a couple of different markers, both at the protein and mRNA level. So you can see, for example, for CD95, in general, you have high expression at the protein level compared to mRNA. And then for CD7, you actually see slightly different dynamics where for protein, you see the highest expression at 1 week, but for mRNA, you actually see earlier activation, and it kind of tapers off. Down here, I'm showing you biplots of, on the X is TIM3 protein versus mRNA. So if you look at protein, you'll see that upon stimulation, you have upregulation of TIM3 protein expression, kind of throughout the course of the activation. But if you look at mRNA, then you'll see that it slowly increases from 3 days to 7 days to 14 days, and then it actually goes down if you look at the transiently stimulated sample. Another, I guess, application that we're really excited about is using single cell, basically BD Rhapsody and AppSeq, and in conjunction with flow cytometry to do biomarker discovery. So this is one example where we identified a gene. So this is looking at mRNA expression that TNF-SF14, which is a TNF family ligand, it's specifically upregulated in the day 3 samples and not really at any other time point, indicating that it's probably important for early activation. But we didn't have this marker in our original AppSeq panel. So we later on went and did a flow experiment, and you can see that it correlates really well with what we see in mRNA. So this is an example of a workflow where you can do a single-cell RNA-seq experiment, identify potential biomarkers, and then use flow cytometry to validate and then process larger numbers of samples. We've also spent, as I mentioned, a lot of time developing new assays for our platform. So there are, in terms of RNA sequencing, there are 2 approaches. What most people are familiar with is a whole transcriptome approach, where it's unbiased and you're looking at everything. But some of the downside is it's much more costly because you're looking at everything, including all the high-expressing housekeeping genes. But we understand it's a really important tool for discovery. This is our, I guess, our approach to whole transcriptome assay, where we use random priming and extension to basically create the whole transcriptome library. And this is a very simple workflow. In comparison, we also have the BD Rhapsody targeted assay, and alongside it is a host of fixed panels that we've developed and validated internally, but there's also custom panel capabilities. And this is really a straightforward assay where, following cDNA synthesis, we do 2 multiplex PCRs, so these are nested PCRs, and a final indexing PCR to generate the final sequencing library. And some of the advantages of doing targeted versus whole transcriptome, as you may know, is first cost efficiency, but also sensitivity. So this is an experiment where we are looking at PBMC cells, and with the same sample, same Rhapsody run, we generate a whole transcriptome library as well as a targeted library. As you can see from reads per cell, we're using about a small fraction of the reads per cell for the targeted library to get slightly lower but comparable sequencing depth. But even with the lower sequencing depth, if you look at dropout rate, you'll still see higher sensitivity in the targeted data. So the dropout rate here is looking at, for example, percentage of CD3D expression detected in CD3 cells. And these cell types are characterized based on AppSeq markers. So across all the ones that we looked at, you have much higher detection of these cell lineage markers in the cell types that you expected to be expressed in compared to whole transcriptome. So the workflow that we think will be really useful going on in the future as people start to adopt single-cell technologies and want to process more and more samples is this whole transcriptome analysis to targeted workflow where, let's say you're starting out with a new model system or a new cell type, you can do a whole transcriptome assay to identify the genes that you're interested in. And then following that, you can design a targeted panel that will allow you to perform more routine single-cell experiments at a lower cost and higher sensitivity. And we've developed a pipeline, a bioinformatics pipeline, to take whole transcriptome data and design primers so that it's sample specific. So as you know, there could be different polyA sites that are used between different sample types, different cell types. So we've devoted a lot of resources into building that pipeline to enable this workflow. So what's next? As I mentioned, we released the WTA protocol end of last year, and we are going to be launching a kit sometime this summer. And we're also working with SeqGeek to expand a lot of the capabilities that they have, so building new plugins, making sure that the software is user-friendly, that's compatible with a lot of the Rhapsody pipeline outputs, and just building new tools. And it'd be great to get feedback from you guys on what types of tools and analysis features that you would love to see. And finally, we're currently developing a VDJ assay. So this is to do T cell and B cell immune repertoire profiling. I'm going to show you just a preview of some of the internal data that we recently generated. So the idea is that we want to be able to harness the power of single cell technologies, looking at mRNA and protein information, and then tack on clonality. So this is just a very straightforward donor PBMC data set where we did Rhapsody targeted assay as well as AppSeq, and then looking at the immune repertoire in addition. So up here, this is looking at CD4 protein expression. So you can see this is your CD4 population, this is your CD8 population. We have CD485RA in the panels to differentiate between the naive and memory populations. And then this is showing actually the top 8 clones that's identified in this PBMC sample. So this is just a healthy donor, so you don't expect to see a lot of expanded clones, but even then you can see that for the clones that are most highly represented, they're more enriched in the CD8 central memory population, which is what you would expect. So this is just a sneak peek of what we're working on. We're really excited about this assay and just the ability to include that into our portfolio and enable immunologists to not just to be able to pair expression with clonality. And we think it will be a very powerful tool. So in terms of future and challenges, I think that this is a growing field. It's a very new field. We're still looking at how to apply this technology, but also looking at how we can overcome some of the limitations. So for example, understanding what biases we're integrating into our data, especially when it comes to, there are different levels of biases, but when it comes to whole transcriptome analysis, different assays will have different GC bias, there are different amplicon length bias. These are some things that are not always talked about when choosing a technology. Another is, as we expect to use more and more of this technology to look at different biological questions, but eventually also moving into the clinical space, there needs to be development of control reagents so that we can compare between experiments, even across platform. And there's been a lot of really cool work done on the informatics side, developing algorithms to eliminate batch effect to allow even comparisons between, say, human and mouse samples. But another question is, with single-cell technology, at least with single-cell RNA-seq, it's important to have a single-cell suspension. So that requires disassociating your cells and potentially storing them. And there's not a lot of studies that looking at how these different disassociation methods or fixation methods or just even storing them on ice and how that affects gene expression and protein expression. So that's something that requires more study to understand so we can better design experiments. And as I mentioned, I think there's a great need for tools for analyzing single-cell multiomics data. As more and more people adopt this technology, not all of us are computational biologists and are comfortable with coding. So we're really dedicated to providing tools that are user-friendly and will enable the biologists to analyze their own data. And finally, cost efficiency. This is a big reason why we are focusing on the targeted assay in order to provide a way that is cheaper on the sequencing side, but also provide higher sensitivity in the assay.